Hello and welcome. Hope everyone's having a fantastic reInvent. I'm Margot McDowell, Principal Product Manager for AWS HealthOmics, and I'm here today with Mirko Buchholzer, VP of Software Engineering at Natera. So today we're going to kick things off with a patient story. Then I'll give a brief introduction to AWS HealthOmics, one of the services that Natera is using today. Then I'll hand things over to Mirko to talk through how Natera is scaling clinical diagnostics operations using AWS services. And then we'll close things out by talking about a few new features and capabilities that AWS has recently launched that might be of interest to other customers in the healthcare and life sciences space. Let's get started.
I kept going to doctors, and because of my age, they thought I had pudendal neuralgia because I had a mountain bike wreck, and it was a huge pelvic trauma. And then one day, my urologist was out of town, and the substitute urologist basically looks at me and he goes, "Have you ever had a urine cytology test done?" And he goes, "It's a simple P-cup test. It checks for atypical cells in your urine. It could reveal cancer." I go, "Great, let's do it." Came back as atypical cells. A few minutes after that, I was looking at the tumors inside me. And that's all it took. They removed the tumors from my body, they biopsied it. We found out that I had stage 3 bladder cancer, and I was gonna be going on chemotherapy. I have a really great oncology team. So I have one oncologist who's kind of like the quarterback. He is on the cutting edge of everything, and he's the one that found Signatera for me. We started doing the Signatera testing pretty much as fast as we could. The Signatera test started to show that the cancer incidence was rising, basically. So I lived for about a year watching these Signatera results kind of ping around, but they were steadily rising. So my doctor, this was new to him, he goes, "I think we're gonna have to do a PET scan." So we did a PET scan in January of that year, and the PET scan lit up in my limbic system near my neobladder, and it showed that there was some activity. And he goes, "I think your cancer's coming back." And the craziest part was, because Signatera had found the cancer so early, the lymph nodes were so small, they were 12 millimeters in diameter, which is cancerous, but it was right at the border that he goes, "Really?" And he needed more information, so he was given the PET scan results. They did another CT scan, and he goes, "Okay, yes, the cancer has returned." Natera caught it so early that the radiologist was like, "Are you sure this guy's got cancer still?" Signatera gave me a 9-month head start, basically. We found my cancer, put me back in remission all 9 months before traditional CT scans would have found it. It was incredible. And now I've just been on a surveillance regimen and immunotherapy maintenance regimen, which again, I get to end pretty soon. And Signatera's getting that long view for me and making sure that I could live my life with my family and just be comfortable. Because if it does come back, I've got so much time. I've got time to make a decision, and I don't have to worry so much. There's a lot of synchronicity actually in my life with cancer. So with my career, I work in radon mitigation supply, testing and mitigation, and I see the same trepidation. With radon, for years and years, you only had short and long-term tests. These you would put in your house for anywhere from 2 to 4 days, or sometimes 90 days or a year, but it gives you one number. And this takes dozens of data points within the first hour of it being plugged in. So to me, it's like, this is like a traditional CT scan, and this is like Natera testing. You're still getting reliable data, but you're getting much more of it on a much more microscopic level. And to me, I'd rather have more data.
He's still been doing immunotherapy, which can be hard on the body still, but his Natera tests are still showing no cancer, which is, I mean, so exciting. I don't know, it brings peace of mind, being able to, just being able to know, especially with a family.
Natera is a global leader in genetics and diagnostic testing. They offer more than a dozen diagnostic tests in the fields of oncology, women's health, and organ health. Natera has performed more than 10 million diagnostic tests since they were founded. They've published more than 100 peer-reviewed articles, and they've been issued hundreds of patents. Natera is considered a leader for innovation for developing tests like Signatera, a test that has already helped more than 250,000 patients like Brent. Because of its sensitivity and effectiveness, Signatera has become one of the fastest growing genetic tests in the world. And in order to keep up with the incredible growth, as well as a portfolio of other growing tests, Natera needs solutions. that help them scale without compromising on turnaround times, that also help with collaboration so that their teams can focus on innovation. And lastly, so that their teams can focus on the science and not the underlying infrastructure. So here at AWS, we talk to a lot of customers in the healthcare and life sciences space. And after talking with clinicians, researchers, and bioinformaticians, we found that they can actually spend a pretty incredible amount of time standing up bioinformatics infrastructure, managing and maintaining ETL for common genomic data sets and data types, and doing a lot of really repetitive work just to get to that first analysis. And because of this, we built AWS HealthOmics, a service specifically designed to accelerate scientific breakthroughs with fully managed biological data stores and fully managed workflows. Customers are using HealthOmics today in 3 key ways. The first is production scale clinical diagnostics tests, similar to Natera. These are tests that can screen and monitor patients for disease and help them with their treatments. Also, we have customers today who are using HealthOmics as a place to experiment with generative AI and experiment with biological foundation models to build full end-to-end drug discovery pipelines. The service can be used to test out biological foundation models or BioFMs, which are a type of advanced generative AI model trained on biological data. Then you can orchestrate an end-to-end pipeline. So for instance, you could have one BioFM that does protein folding to predict protein structure. You could have another that generates small, novel, drug-like molecules. And then you could use a third BioFM to actually look at the interaction between those two. And then iterate over that workflow or that pipeline hundreds of times, thousands of times, or even millions of times to screen for drug candidates. And lastly, we have customers today using HealthOmics for multimodal analysis. So genomics data on its own is incredibly powerful. It becomes even more powerful when you can combine that with other data sets. So HealthOmics helps customers today transform their variant data, which is semi-structured, into structured data, and then combine that with other data modalities in order to give a more comprehensive view of patient health. So let's dig into how it works. We intentionally designed the service in a modular fashion. So we wanted customers to be able to adopt individual modules or components of the service, integrate that into their existing solutions to get started quickly. For customers using all the components of our service, their architecture may look something like this. So first, you can bring in raw sequence data and store that in the sequence store. We also offer a reference store for reference genomes. Next, you can run analysis on that sequence data using bioinformatics workflows. Those can be defined in different common bioinformatics languages or domain-specific languages. And the output of a lot of those analyses tends to be something called a variant calling file or a VCF file. So we also provide a solution for that. We can take semi-structured VCF data and we can store it in a format in a variant store that's queryable. We also provide an annotation store, which allows you to bring other data that can give context to your variants. And then you can query and analyze that data using other AWS services like Amazon Athena and Amazon EMR so that you can combine it and analyze it alongside other data sets. So we built the HealthOmics sequence store specifically because we heard from customers that storing petabyte scale genomic data is a challenge. It's hard to do it cost effectively. It's hard to do it in a way where you can find all of your data sets, and managing permissions is also a challenge. And so we developed the sequence store to allow customers to bring in petabyte scale data to be able to catalog that data in a way where everything is discoverable and findable, and you can attach domain-specific metadata, like a subject ID or a sample ID to keep all of your data very organized. Then we offer automated genomic-specific compression. So when you have data that you're bringing into the sequence store, We store it in a compressed format so that you get the best possible cost for saving that, for storing that. And then when you want to access that data, you can reactivate it and make it available for usage. We have built-in e-tags for data provenance, and then also sharing and collaboration features. So with fine-grained access controls, you have the ability to share only the specific data sets required with specific individuals. To get started with a sequence store, you can import FASTQs, BAMs, and CRAMs. These are common genomic data sets or files. And when you kick off an import job into the sequence store, you can attach metadata, like a subject ID or sample ID at that time. And HealthOmics also creates a unique e-tag so that you can uniquely identify every object in the sequence store. And then you can integrate that sequence store into your applications using either HealthOmics or AWS S3 APIs. So you can use genome browsers or any custom tool that uses SAM tools or Botto3, for instance. Additionally, the sequence store is integrated to work with HealthOmics workflows for analysis. So we built HealthOmics workflows specifically because we heard from customers that it takes a lot of work to get to your very first analysis. So you have to manage EC2 instances, you have to spin up file systems, maybe resize them, and then there's the actual workflow engines themselves. And those workflow engines get upgraded regularly, they need to be patched, they need to be maintained. And if your organization uses multiple bioinformatics languages, it means that you need to maintain multiple engines. So this can really take quite a bit of time and resources and maintenance just to maintain the underlying infrastructure. So we built HealthOmics workflows so that our customers could start runs with a single API call. So all you need to do is build your analysis, and then you can call start run. And HealthOmics will provision the underlying infrastructure. It will provision the underlying infrastructure exactly in the amounts of vCPUs, GPUs, and memory that's requested. And when you're ready to scale, all you need to do is call start run more times, and the HealthOmics service automatically handles all of the scaling on the back end. This gives our customers a predictable cost per sample, pay as you go, so you don't have to pay for persistent clusters when you don't need them and when you're not using them. And really allows customers to think about the actual analysis itself. So workflows are available in two forms. The first is private workflows, and the second is ready to run. Private workflows allow customers to bring their own custom written bioinformatics scripts in WDL, Nextflow, or CWL. And then you can bring your own containers, which should be or gives you the ability to bring your own tools, packages, dependencies for your workflow to use. But if you prefer to get started quickly and you don't want to write any code, we have an option for that as well. Our ready-to-run workflows are a catalog of around 37, or we have 37 workflows today. Everything from AlphaFold, ESMFold, GATK, NF Core, all pre-built so that you don't have to write code to get started. And we've also partnered with a number of partners in the industry, Sention, NVIDIA, Ultima Genomics, and Element Biosciences to also bring their analyses to HealthOmics. Once you start a run and kick off your workflow, HealthOmics will provision a separate EC2 instance for every task and a file system for each run and manage all of the networking and the actual engine node that's orchestrating the full end-to-end workflow. So a common output of workflows are variant calling files. And variant calling files, VCF files, can take a bit of work in order to be able to compute on them. So the data is semi-structured. So the first thing you need to do is basically build ETL to transform that data into a format that's easier to use, easier to query. And you also need optimal partitioning if you want fast performance of those queries. So there's some structuring of that data that's very important. And then lastly, in order to give context to that data, you need annotation data. And you may have multiple versions or types of annotation data that you want to use. And so our HealthOmics variant store is designed to make this a zero ETL process for customers. You bring a file, you run an import job, and the variant store takes care of all the automated data transformation and stores that data in a format that's optimal for fast queries. There's also built-in sharing and collaboration features. So you can use lake formation to control row and column level permissions. And the variant store integrates deeply with AWS analytics services. So you can use Amazon EMR, Amazon Athena, QuickSight, and a variety of other tools in order to query your variant and annotation data. So the HealthOmics variant store, as I said earlier, like the genomics data on its own is very powerful, but it becomes considerably more powerful when you combine that with other things like clinical records and imaging data. And so we have a couple of other AWS services that help with that today. You can use AWS HealthLake to bring in clinical records. You can use AWS Health Imaging to bring in imaging data alongside your variant store. And then you can query all this data together to give you a more comprehensive view of patient health. HealthOmics customers are operating at scale today. They're running thousands of samples daily, tens of thousands of concurrent vCPUs and tasks with our HealthOmics workflows. They're also storing hundreds of petabases of data. For instance, UK Biobank is democratizing secure access to whole genome sequences of over 500,000 volunteers using the HealthOmics sequence store. And other customers tell us that HealthOmics helps them to save an incredible amount of time and accelerate their research and development efforts. For instance, Roche cut time to complete cancer research down from 1 year down to 3 months. Sonrai Analytics, a drug discovery startup, cut their drug discovery research times down by 70%. And the Children's Hospital of Philadelphia can now access multimodal data sets within minutes instead of months. And lastly, Natera is using HealthOmics workflows for their Signatera diagnostics test and is part of their internal cohort builder using our variant stores. So with that, I'm going to hand things over to Mirko to talk through how they are building on AWS.
Hi everyone, my name is Mirko Bolzer. I'm the VP of Software Engineering here at Natera. It's always grateful to be here at reInvent. It's one of my favorite events in the whole year. A lot of great minds are basically coming together and are helping us to kind of think about new things on what we can apply to our companies in the coming year. So for me, being here at reInvent is always a very great thing to do where I can think about new ideas, new things that I want to apply next year. Today I'm here to talk about Natera itself. We had quite a bit of a journey in the past couple years. Natera was founded in 2004, has pioneered the cfDNA technology across the spectrum. We're using this technology to detect fetal tumor and donor DNA and are basically able to do that with a single blood draw without any other interventions and can run different tests across the spectrum. Our laboratories are based out of San Francisco in the Bay Area, as well as in Austin, Texas. And we just recently passed over 10 million tests that we have performed worldwide. Our aim is really to transform the management of disease worldwide. This is a bold statement, and to be able to do that, we need to invest in many different aspects around the whole organization to be able to do that. Scientific innovation is a big component of this, but scientific innovation alone is not going to help the patient which needs the help right now. So with that, we're basically working on fast innovation cycles and making sure that we can bring more tests and more patients and can help more patients day in, day out. I think Brent's bladder cancer journey was a big message there and very touching for me each time when I look at it, and is motivating me every day to get up and help improve kind of like the change of disease. So this is kind of like a test that Natera has introduced in 2017. A test that has changed cancer treatment quite a bit. We have over 250,000 patients which are using this at least one time, and most of them are on a monitoring regimen. We support over 50 solid tumor types and are able to leverage the Signatera test across different classes of cancer. The Signatera test works really in a basis for residual disease detection. So when we're working with a patient, we're getting a tissue sample that we're then sequencing, where we're detecting particular mutations of that tumor tissue sample, and are then creating a patient-specific fingerprint for that tumor that we're using for the monitoring assays. If the monitoring is happening, we're basically getting blood samples over time. In general, we should, after treatment, get less and less residual disease that we're seeing within the test itself. So the goal is to get to negative. The test can not just be used for the initial component, but it can also be used later on for monitoring. So recurrence is a big aspect of cancer journeys with patients, where we're constantly monitoring the patient and making sure that the cancer doesn't recur. So in this case, we can even detect recurrence up to 9 months earlier than standard imaging, which I think personally allows the physician as well as the patient to get treatment early on, and we can manage the cancer way better. Signatera is a pan-cancer test. To be able to prove that, we basically were running quite a bit of studies around colon cancer, breast cancer, lung, and bladder cancer. As you can see on the screen, each of those studies were basically showing that the sensitivity and specificity of the actual test was pretty similar across the different cancers. This means we can use the same test across all the cancer types and have a pan-cancer capable test that is leveraged in multiple settings. Natera doesn't only do oncology testing. In fact, Natera initially started with women's health testing. From planning a family to prenatal testing to hereditary cancer screening, everything is available on the women's health side, where we can basically work through many of the diseases or planning that we're doing on the family side. Same thing on the organ health side, where we have tests which have the ability to assess otherwise undetected rejection events that might lead to transplant organ failure. So this is kind of like key areas that Natera is currently working on, but the goal really is to expand this to different other areas as well, and being able to transform the disease management across the spectrum. So if we want to do this, I think we kind of like have 3 core components that we need to invest in to be able to do that and scale that. Obviously, scientific innovation is a core aspect of it, being able to find new tests, being able to go through kind of like new insights, being able to leverage our vast genomic and clinical data is a key aspect of it. Scientific innovation doesn't help the patient right now if we're not able to actually bring it to the market. I think what we're seeing a lot in the healthcare industry is great scientific breakthroughs which take then decades to be able to being actually implemented with physicians and patients. So another aspect that we're trying to really push is the ability to bring those tests to market very quickly. In the past 5 years, we have been releasing 8 tests to the market in different fields, as we have seen just previously. And we think that we need to have a flexible and scalable infrastructure to be able to do that in grand scale. The other aspect, as Margot mentioned, Signatera is one of the fastest growing genomic diagnostic tests. And with that scale, we need to have the architecture and infrastructure available to actually execute those. It is very relevant for specifically Signatera tests to have results very quickly back to the patient, which can then, where the physician then can take the different strategies for treatments based on the results. To be able to implement that, we kind of rethought about 2 years ago on how we want to structure our lab. We basically went on an adventure to create a digitally connected lab. We call it LabX. So the goal here was really to create a scalable infrastructure that we can scale across different locations, different tests, different areas that we're testing, where we have large lab automation implemented and where we have reusable infrastructure as well. So currently we're processing over 54,000 tests a week in two labs. This results into about 60,000 workflows that we're executing, and we're generating about 1.6 petabytes of data in 1 week. When we're looking at this, we looked at the structure of how we can actually implement this. So we have 2 areas where we focused on. One is the digitally connected side, which is running on AWS cloud, where we're having many of our services integrated into our cloud services as well as AWS HealthOmics. The other piece is the lab component, which basically is focused on the automated work cells that are executing some of our assays that we're running through the lab in the wet lab workflow, and then have accordingly the lab equipment where our sequencers and other lab equipment are generating the data that is then pushed back into our cloud infrastructure. A key component that we introduced while we were doing this redesign was really around assays and the assay execution. So a lot of labs are basically just running particular products in their labs on different lines. We were thinking of changing this quite a bit in regards to being able to reuse assays across different products. So an assay consists of a wet lab workflow. It consists of a bioinformatics workflow and then has a QC step to validate the results of that particular assay. This then gets aggregated on the interpretation and reporting side, where we have the ability to then create the end result and the report. So if an order is coming in for a particular product and we're accessioning our samples, this then gets mapped into the different assays that need to be executed. So one product might have one or more assays that will be executed, and going through the whole lab workflow, bioinformatics workflow, QC, and all those different assays aggregated into the interpretation reporting layer. So there we have the ability now to reuse those assays across the different products. So we can have certain assays which are used particularly in many of those products specifically. So the other component here is the ability now to also batch and share samples across product lines as well. So we have the ability to just run the assay no matter what product is actually executed on top of it. All of this gets then connected through our event bus and our storage system that we're using to connect all of this. And on top of it, we basically have the lab analytics component where we're pushing not just the genomic data and the results and the assay QC metrics, but also have corresponding lab metrics that we're using to optimize the lab directly. So this gives us kind of like a new structure, a way on how we can scale the lab, and the way on how we are kind of like have the reusability of the assay itself. So to visualize that a little bit, I created a little animation here where you can see on the left-hand side basically the products that we're running, and on the right-hand side you can see some of those assays that we have in the catalog, which are basically representing either a wet lab workflow, a wet lab workflow plus a bioinformatics workflow, or a bioinformatics workflow only. Each of those products are using different assays when they get executed. So for example, Signatera, if we have a second time point here, we might run an NGS workflow with a corresponding pipeline, and then do, for example, a tumor normal comparison and figure out what the actual output is. Same here for Renasight, might use the same NGS workflow, have another workflow as well, and then for confirmations, for example, we might use an RNA assay that we're executing on top of it. And then if we go to Horizon, for example, leveraging the same assays, but now has a particular assay which is specific for screening workflows. So this allows us to kind of like run assays within the lab. The lab is really focused only on the assay execution, doesn't really matter about which product is running it. And we can also do cross-product sample sharing very easily. So LabX is using HealthOmics to be able to scale. We were looking at solutions that we can implement to really scale our labs to the next level. HealthOmics was a good solution for us. We're running all of our bioinformatics pipelines via WIDL on HealthOmics. We standardized on WIDL, which was also a great choice in regards to simplifying some of those steps that we have within the workflows. So those workflows can be quite complex, and we have reusability within the workflow as well. All of this is Docker containerized, so we can run those in particular versions, have full version control across the bioinformatics workflows as well. The other piece is the scalability. So one API call or one event allows us to really execute the full bioinformatics workflow. HealthOmics takes care about the orchestration of the workflow engine itself, takes care about the compute, the file system, and the final storage as well. So this kind of gives us an easy way to run those workflows and scale them up. The other components that we're using are really around the VCF annotations. So those are components that we are leveraging as well, together with the variant store, where we're storing all of those VCF variants and using them to do cohort building, as well as general queries for QA or R&D. We touched a little bit on the analytics platform as well. So we saw during the actual LabX components, there's the analytics component where we're pushing certain data sets into the analytics data mesh. It was key for us to have a distributed data store, which allows us to have each single component and application team being responsible about the actual data that is pushed really diverse in each of those sections. And then we have a central governance as well as a central catalog, which allow us to have easy access to the data, but in a secure, compliant way. So some of those metrics that we're pushing there are around pipeline outputs, instrument data, or lab metrics that we want to have in there to optimize the lab itself. The other components are really around other clinical data that we're going to touch just in a bit. This data is then used in many different ways, from simple data quality assurance to interpretation to lab operations, where we can start to optimize our lab workflows and labs in general. The other component is this is a great store for our R&D teams and also for our data products to create new data sets on top of what we're getting into the data mesh from the different lab components. So one of those other data sets is really around the clinical data that we're receiving through either a sessioning where we have path reports or other documents from the patient, or integrations that we are doing into EMR, and being able to extract clinical attributes from those unstructured data sets and store them as well into our product catalog. The way on how we're doing this is extracting basically the core text out of those documents. This could be done through OCR. Some of those documents are basically already containing the text itself, so we just need to extract the text and then push this into our vector store. So we're using LLMs to create vectors of those data chunks and storing them into our embeddings vector store. From there on, we can run workloads on top of it, which allow us to extract clinical data based on prompts that we're using with the LLM. So LLMs are now able to extract the right data from those unstructured data sets. We can run through validation and then ingest that into our data mesh and have a full patient health record that is available there for us to use. With that data, we can combine it now with the clinical data with our genomic data, as well as our results. So if we're looking at this, we can generate full patient cancer journeys. So in this case, we have a stage 2 bladder cancer patient. And on the screen, you can see on the left-hand side is basically non-invasive surgery that was done on day 0, and then a neoadjuvant chemotherapy, which was applied between day 52 and 94. Later, the physician decided to actually do an invasive surgery as well, because it was growing. So with that, we got a tumor sample, and we're able now to run Signatera with that tumor sample. So we created the fingerprint of the tumor, and we're now able to run monitoring across the time. And you can see after that surgery, the first 3 time points were basically negative. Time point 4, though, showed a 0.07 MTM value already, And for Signatera, that moved it into a positive stage. And you can see 4, 5, 6, basically the MTM value was going up, which basically showed that something is going on within that patient. And on day 573, basically the physician declared that there is a distant recurrence and we need to change the actual treatment. So radiation as well as immunotherapy got applied, and you can see the following data points basically move back into negative. So this was basically successful for this treatment, and the patient stays on monitoring. So now if we combine our data, the results as well as clinical data together, we can now create those type of cancer journeys for each of our patients. And obviously we can combine those as well. So what you're seeing here on the screen on the horizontal lines is basically a cancer journey for each patient. So you see kind of like indicators about the Signatera results, indicators about surgical procedures, therapies, and then also outcomes. This helps physicians to really compare their patient with other patients, helps them to look at treatment timelines, but then also allows them to figure out treatment strategies based on other patients that they have in their clinic. So with this, you can really get an informed approach on the patient care. So to reflect all this, I think we really want to change the way on how we're doing care across the different spectrum. To get that going, we have to do rapid innovation. And scientific innovation and breakthroughs is one aspect of it, but being able to actually get that down to the lab and being able to execute that day to day with patients is another aspect. So for us to be able to do that, we think that reusable digitally connected lab infrastructure enables us to scale that. And with that, we have the ability to bring tests to the market way quicker. Having access to distributed data is a key aspect within the organization to create new insights across genetic information, but then also clinical information. And last but not least, the new abilities with generative AI and the ability with new LLMs, we have now the capability to extract unstructured data out of the clinical data that we're getting out of EMRs. I think this will transform the way on how we're going to work and is helping us even more to get new insights and abilities to bring new tests to market. It is very rewarding to get up every morning knowingly that you can help a small batch of patients with the work that you're doing. And I'm pretty sure a lot of you in this room also have the same experience. So thank you, everyone. And I'm going to hand it over to Margot to talk about a couple new features.
Awesome, thank you, Mirko. Okay, so I wanted to leave us today with 2 new features that are new capabilities AWS has recently introduced. One is for anyone here who has developed new workflows before. Just a few weeks ago, AWS HealthOmics introduced support for call caching. So call caching is the ability to resume runs from the last successfully completed task. So essentially, when you're developing new workflows, involves writing code. When you're trying that out for the first time, it's pretty common to have syntax errors or other issues. And so being able to pick up from the point of a code change or from the point where your workflow has hit an issue really helps you speed up workflow development. So HealthOmics is now intelligent enough to figure out exactly when you give it the same file, the same task definition, the same set of containers, we can store or cache task results and then be able to reuse them so that you can skip over things that have been computed before. So this saves a ton of time and also accelerates new pipeline development. So we're very excited to introduce this into HealthOmics workflows. Also, the actual run cache itself is S3 based, so we can scale that with your data. So you don't have to resize your cache or keep increasing the size. You can store as much data as you need to. And we've introduced so direct access to that cache. You can use those files for advanced troubleshooting and debugging. And we also have a new set of CloudWatch logs that help you analyze this data. So we're excited about that. The other thing I wanted to talk about is digital pathology using AWS HealthImaging. So one of the other services that we offer is HealthImaging. It's a service that allows customers to store, analyze, and share medical imaging data at petabyte scale in the cloud. And so it can lead to considerable cost savings by allowing you to store only one copy of an image and then be able to access that with sub-second image retrieval latencies. So customers use HealthImaging today for X-rays, CTs, MRIs, ultrasounds. But we're pleased to introduce that HealthImaging now offers enhanced support for lossy medical imaging data. So this enables support for DICOM whole slide images. So if you operate a laboratory today and you have pathology operations where you take your samples, mount them on a slide, you can take them through a slide digitization process, and then actually send that image now to AWS HealthImaging. So your pathology on-site or from anywhere in the world can go access these images at very, very low latency. And when combined with other services, you can do some pretty incredible things. So in this example, you can de-identify the image and then run an Amazon SageMaker model on it. So you can detect and kind of analyze things in that sample. And lastly, with this data in AWS HealthImaging, you can more easily combine it with omics data or other clinical data to give you an even more comprehensive view of a patient's health. So just to recap, we talked about a few things today with HealthOmics. It's a service that helps you to scale clinical diagnostics pipelines as well as drug discovery pipelines. And it offers customers a predictable cost per sample and fast turnaround times. It has built-in sharing and collaboration features as well as fine-grained access controls so you can keep your data secure. And also, it allows our customers to innovate faster with fully managed biological data stores and fully managed compute. So if you'd like to learn more about HealthOmics or HealthImaging, check out our QR codes. These will land you on our product marketing pages. It'll tell you a bit more about both. And if you'd like to hear more Natera patient stories, check out this last QR code. And I wanted to say thank you to everybody for joining today. And I hope everyone else has a great rest of your reInvent. And please fill out our session survey. Thank you.